plant structures.
Okay, now we're working on the plant structures lab, and we took a carrot and we cut it in as thin slices as we possibly could, and then we used the micropipette to apply the iodine. Now, I can't see anywhere in the lab where it explains this, but iodine solution is an indicator solution. It turns blue in the presence of starch. So the mere fact that we are seeing that the dye is taken up into the plant material is an indication that this is a starchy storage, or again, it's a carrot, it's a root. And then we are going to look at it on a slide stained, and we also prepared one that's not stained just so we could see what the difference is. Now, hold on one second. So what we hope to see, which you might see or might not, but please try, is that the cambium, the inner part, and the xylem on the inside, would take up less starch, and then the phloem, which has sugar in it, would take up more starch, and then the cortex. So we might see a variation in how much iodine was taken up by the various parts of the cross-section of the carrot. Here's like another example that kind of shows that in a little bit more color, but with less diagrammatic information. No. Now we're doing the celery part, right? And it says to use like a half a bottle of food coloring, but really we just need to cover the bottom because you want to get a nice flat, wait, hold on, maybe we should fix this. We want the flattest edge possible so that the cross section of the celery is flush to the water that you put into the cup. Because see there, you can see the vascular tissue. So we want that vascular tissue in the food coloring. You don't need to use a whole bottle to achieve that. So hold on, we'll fix our cut. Let's put our food coloring in the jars, and we'll get that coating on the bottom, and then we'll try to sink the vascular tissue right into it. I didn't even know they made black food coloring. And so here we're measuring transpiration, and essentially we're trying to prove to ourselves that plants drink water from the ground up, right? And they produce food products from the leaves, but we're not working on that right now. We're just watching the transpiration process. We're watching how the water is going to defy gravity. And we're going to know it's defying gravity because the celery will change color as the colored water moves up through the plant parts. And then another part of this lab is to look at the tree cookie, which comes in your lab kit. But I'm struggling with this one because what I can see more prominently is the grain from the cut. See, there are some concentric circles in there, but we're seeing these vertical cuts. So if you have time and you want to make this into something that is a memento forever, sand it. And then let's look at a picture on the internet. Okay, so here's a tree ring picture I just found on the internet. But see, now you can really see what you have to label for the lab. So you can see the growth rings, and remember that in the winter, the tree is putting on a thinner, darker ring, and in the summer, depending on moisture availability, it's putting on a thicker ring. So that's how we can tell that these are annual rings. And then you can also see in this particular cookie how, look at how this plant became more drought stressed as time went on, because those rings are wider towards the center, which represents when the tree was younger. But as you go out to the last, decade or two, those are exceedingly thin, which just tells me that there was reduced moisture availability in those years.